everyone. Today let us discuss about carbohydrate metabolism. So first let's come to introduction. Carbohydrate metabolism denotes the various biochemical processes responsible for the formation, breakdown and interconversion of carbohydrates in living organisms. The most important carbohydrate is glucose, a simple sugar monosaccharide that is metabolized by nearly all known organisms. Carbohydrates serve as energy sources and as structural elements in living cells. The metabolic pathways of carbohydrates are: first, carbon fixation or photosynthesis, in which carbon dioxide is reduced to carbohydrates. Second is glycolysis. It is the oxidation metabolism of glucose molecules to obtain ATP and pyruvate. Third is the Krebs cycle or citric acid cycle, in which pyruvate from glycolysis is further broken down in aerobic organisms. Fourth is glycogenesis, the conversion of hexoses into pentoses, and in NADPH regeneration. And fifth one is glycogenesis, the conversion of excess glucose into glycogen. And sixth one is glycogenolysis, the breakdown of glycogen into glucose. Seventh one is gluconeogenesis, that is the de novo synthesis of glucose molecules from simple organic compounds. So let us come to glycolysis. Glycolysis, it comes from the Greek word glykes, meaning sweet, and lysis, meaning splitting. It was discovered by three German scientists, Gustav Emden, Otto Meyerhof, and J. Parnas in 1930. Hence, it is also known as Emden-Meyerhof-Parnas pathway. It is common to both aerobic and anaerobic respiration. So let us come to the definition of glycolysis. Glycolysis is the conversion of glucose molecule into two molecules of pyruvate through a series of 10 enzyme-mediated reactions to form the high-energy molecules ATP, adenosine triphosphate, and NADH2, which is nicotinamide adenine dinucleotide. So the site of reaction of glycolysis is, it occurs in cytosol or cytoplasm. The entire glycolysis pathway can be separated in two phases. First is the preparatory phase, wherein ATP is consumed, and second is the payoff phase, which yields ATP and NADH. So first let us come to the preparatory phase. So in the preparatory phase, let us come to the first step. So in the first step, it is phosphorylation of glucose. Glucose is phosphorylated to glucose 6-phosphate by ATP in the presence of the enzyme hexokinase and magnesium ion. The reaction is irreversible. Second is the synthesis of fructose 6-phosphate. Glucose 6-phosphate is isomerized to fructose 6-phosphate by the enzyme phosphohexose isomerase. Third is the formation of fructose 1,6-bisphosphate. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate in the presence of the enzyme phosphofructokinase 1. This is an irreversible reaction. Fourth is the cleavage of fructose 1,6-bisphosphate. The enzyme fructose 1,6-bisphosphate aldolase, often called aldolase, cleaves fructose 1,6-bisphosphate to form two different triose phosphate, glyceraldehyde 3-phosphate and aldose, and dihydroxyacetone phosphate aketose. Fifth is the isomerization of DHAP. Dihydroxyacetone phosphate is isomerized to glyceraldehyde 3-phosphate with the help of the enzyme triose phosphate isomerase. So next comes the payoff phase. So in the payoff phase, which is the sixth step of glycolysis, oxidation of glyceraldehyde 3-phosphate. So in this, its molecule of glyceraldehyde 3-phosphate is oxidized and phosphorylated by inorganic phosphate in the presence of the enzyme glyceraldehyde 3-phosphate dehydrogenase to form 1,3-bisphosphoglycerate. NAD is the hydrogen acceptor. It produces NADH. The seventh step is the phosphoryl transfer from 1,3-bisphosphoglycerate to ADP. The enzyme phosphoglycerate kinase transfer the high energy phosphoryl group from the carboxyl group of 1,3-bisphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. This reaction is an example of substrate level phosphorylation. Eighth step is the isomerization of 3-phosphoglycerate. The enzyme phosphoglycerate mutase catalyzes the isomerization of 3-phosphoglycerate to 2-phosphoglycerate. The ninth step is the dehydration of 2-phosphoglycerate. A molecule of water is removed from 2-phosphoglycerate to yield phosphoenol pyruvate, that is PEP. This reaction is catalyzed by the enzyme enolase. The 10th step is the transfer of phosphoryl group from PEP. The last step in glycolysis is the transfer of the phosphoryl group from phosphoenol pyruvate to ADP, catalyzed by pyruvate kinase to form ATP and pyruvate. This step is irreversible. So after discussing the steps of glycolysis, let's come to the net products of glycolysis. In glycolysis, two molecules of ATP are consumed during double phosphorylation of glucose to form fructose 1,6-bisphosphate. In return, 4 molecules of ATP are produced by substrate level phosphorylation, that is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate and phosphoenol pyruvate to pyruvate. 2 molecules of NADH are formed at the time of oxidation of glyceryl dihydrophosphate to 1,3-bisphosphoglycerate. The net reaction is as follows. Glucose, they react with 2 NAD, 2 ADP, 2 PI to form 2 molecules of pyruvate, 2 molecules of NADH, 2 hydrogen ion, 2 ATP and 2 molecules of water. Each NADH is equivalent to 3 ATP. So that the net gain in glycolysis is 8 ATP. So now let us come to regulation of glycolysis. The rate of glycolytic pathway is controlled by the kinetic properties of its hexokinase isoenzymes and the allosteric regulation of the enzymes that catalyze the 3 irreversible reactions: hexokinase, phosphofructokinase 1 and pyruvate kinase. So first, let's come to the regulation of hexokinases. The animal liver has 4 hexokinases. Hexokinases phosphorylate glucose to glucose 6-phosphate. When blood glucose level is low, phosphorylation allows cells such as brain and muscle cells to obtain sufficient glucose. But when blood glucose level is high, cells do not phosphorylate more glucose molecules than required to meet their immediate needs. Next is the allosteric regulation of glycolysis. The reaction catalyzed by hexokinases, PFK1 and pyruvate kinase can be regulated by allosteric regulation. For example, hexokinases are inhibited by excess glucose 6-phosphate and ATP. PFK1 is also allosterically inhibited by high level of ATP and citrate and activated by AMP and fructose 2,6-bisphosphate. High AMP concentration and fructose 1,6-bisphosphate activates pyruvate kinase, while high ATP and acetyl-CoA concentration inhibits pyruvate kinase. So next comes the fates of pyruvate under anaerobic condition. First, let us come to fermentation. Pyruvate occupies an important juncture in carbohydrate catabolism. Under aerobic conditions, pyruvate is oxidized to acetate, which enters the citric acid cycle and is oxidized to carbon dioxide and water. And NADH formed in glycolysis is ultimately reoxidized to NAD by passes of its electron in oxygen in mitochondrial respiration. Under anaerobic conditions, a number of cells and organisms convert pyruvate to a more reduced organic compound such as lactate or ethanol and regenerating the NAD required for glycolysis to continue. So here comes alcoholic fermentation. Yeast and other microorganisms ferment glucose to ethanol and carbon dioxide. Glucose is converted to pyruvate by glycolysis, and the pyruvate is converted to ethanol and carbon dioxide in a two-step process. In the first step, pyruvate is decarboxylated in an irreversible reaction catalyzed by pyruvate decarboxylase, and acetaldehyde is produced. Pyruvate decarboxylase requires magnesium ion and has a tightly bound coenzyme thiamine pyrophosphate, that is TPP. In the second step, acetaldehyde is reduced to ethanol through the action of alcohol dehydrogenase. Hydrogen is obtained from NADH produced during the oxidation of glyceryl dihydrophosphate to 1,3-bis-phosphoglycerate in glycolysis. The overall equation for alcoholic fermentation is glucose molecule, they react with 2 ADP, 2 Pi to form 2 molecules of ethanol, 2 molecules of carbon dioxide, 2 molecules of ATP and 2 molecules of water. So here comes importance of fermentation. Fermentations are used in industry to produce a wide range of commercially valuable organic compounds and food products. Example, alcoholic fermentation is used in brewing industry. A variety of dairy products like curd, cheese, yogurt are produced by lactic acid bacteria, etc. So now let us come to gluconeogenesis. Gluconeogenesis is the formation of new glucose molecules from non-carbohydrate precursors like lactate, pyruvate, glycerol, and certain alpha keto acids, molecules derived from amino acids. It occurs primarily in the liver. Under certain conditions, that is metabolic acidosis or starvation, the kidney can make small amounts of new glucose. Between meals, adequate blood glucose levels are maintained by hydrolysis of liver glycogen. When liver glycogen is depleted, example, owing to prolonged fasting or rigorous exercise, the gluconeogenesis pathway provides the body with adequate glucose. Brain and red blood cells rely exclusively on glucose for their energy source. So now let us come to the reactions of gluconeogenesis. The reaction sequence of gluconeogenesis is largely the reverse of glycolysis. 7 of the 10 enzymatic reactions of gluconeogenesis are the reverse of glycolytic reactions. However, 3 reactions of glycolysis are irreversible in vivo and cannot be used in gluconeogenesis. The first is the conversion of glucose to glucose 6-phosphate by hexokinase. Second is the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1. The conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase. So let's come to conversion of pyruvate to phosphoenol pyruvate, PEP. PEP synthesis from pyruvate requires 2 enzymes, pyruvate carboxylase and PEP carboxy kinase. Pyruvate is first transported from the cytosol into the mitochondria, or is regenerated from alanine within mitochondria by transamination, in which the alpha amino group is removed and added to an alpha keto carboxylic acid. Then pyruvate carboxylase, a mitochondrial enzyme that requires the coenzyme biotin, converts pyruvate to oxaloacetate, that is OAA. Oxaloacetate OAA is then converted to phosphoenol pyruvate, that is PEP, by phosphoenol pyruvate carboxy kinase. The magnesium ion dependent reaction requires guanosine triphosphate, that is GTP, as the phosphoryl group donor. PEP carboxy kinase is found within mitochondria of some species and in the cytoplasm of others. In humans, this enzymatic activity is found in both compartments. The inner mitochondrial membrane is impermeable to OAA. Hence, cells that lack mitochondrial PEP carboxy kinase transfers OAA into the cytoplasm by using the malate shuttle. In this process, OAA is converted into malate by mitochondrial malate dehydrogenase. Malate leaves mitochondria through a specific transporter in the inner mitochondrial membrane, and in the cytosol it is reoxidized to OAA with the production of cytosolic NADH by cytoplasmic malate dehydrogenase. Next comes the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. The generation of fructose 6-phosphate from fructose 1,6-bisphosphate is catalyzed by the enzyme fructose 1,6-bisphosphatase. Next comes the conversion of glucose from glucose 6-phosphate. Glucose 6-phosphatase, found only in liver and kidney, catalyzes the irreversible hydrolysis of glucose 6-phosphate to form glucose and PI. Glucose is subsequently released into the blood. Gluconeogenesis is energetically expensive but essential. The sum of biosynthetic reactions leading from pyruvate to free blood glucose is 2 molecules of pyruvate, they react with 4 molecules of ATP, 2 molecules of GTP and 2 NADH molecules, plus 2 hydrogen ion and 4 water molecules to form glucose, 4 molecules of ADP, 2 molecules of GDP, 6 inorganic phosphate, 2 molecules of NAD. Formation of 1 molecule of glucose from pyruvate requires 4 ATP, 2 GTP and 2 NADH. Hence, it is energetically expensive process. The high energy cost is necessary to ensure the irreversibility of the gluconeogenesis. So now let us discuss about the substrates of gluconeogenesis. Several metabolites are gluconeogenetic precursors. Lactate is released by red blood cells and other cells that lack mitochondria or have low oxygen concentrations. In the Cori cycle, lactate is released by skeletal muscles during exercise. After lactate is transported to liver, it is reconverted to pyruvate by lactate dehydrogenase and then to glucose by gluconeogenesis. Next substrate is glycerol. Glycerol, a product of fat metabolism in adipose tissue, is transported to the liver and then converted to glycerol 3-phosphate by glycerol kinase. Oxidation of glycerol 3-phosphate to form DHAP occurs when cytoplasm NAD concentration is relatively high. Next substrates are the citric acid cycle intermediates like citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate and malate. They can undergo oxidation to oxaloacetate and enter gluconeogenesis. The next substrates are amino acids that can be converted to glycolytic intermediates and undergo conversion to glucose and are termed glucogenic. Alanine and glutamine, the principal molecules that transport amino group from extrahepatic tissues to the liver, are particularly important glucogenic amino acids in mammals. After removal of their amino group in liver mitochondria, the carbon skeletons remaining, that is pyruvate and alpha-ketoglutarate respectively, are readily funneled into gluconeogenesis. So now let us come to the regulation of gluconeogenesis. The rate of gluconeogenesis is affected primarily by substrate availability, allosteric effectors and hormones. Gluconeogenesis is stimulated by high concentrations of lactate, glycerol and amino acids. A high-fat diet, starvation and prolonged fasting make large quantities of these molecules available. 4 key enzymes in gluconeogenesis. It is pyruvate carboxylase, PEP carboxy kinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase are affected by varying degrees of allosteric modulators. Glycolysis is an almost universal central pathway of glucose catabolism, the pathway with the largest flux of carbon in most cells. The glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types, that is erythrocytes, renal medulla, brain and sperm, for example. Fermentation is a general term for the anaerobic degradation of glucose or other organic nutrients to obtain energy conserved as ATP. Glucose stored as glycogen in liver and muscle is not always sufficient during meals and long fasts or after rigorous exercise. The required glucose molecules are replenished through gluconeogenesis. It takes place to maintain the blood glucose level when glucose concentration in the blood is low. Thus, gluconeogenesis and glycolysis are reciprocally coordinated to maintain the energy requirements of our body.